heartfelt gratitude for your presence today, for demonstrating your unwavering commitment to making a national impact. Today, we embark on a new chapter in our journey today toward the activation of technology transfer offices with a global partner. We honored we have to have with us today the researchers and entrepreneurs and expert in the field of commercialization, D.Shad. He's a world-class pioneer who have developed more than 2,000 commercialized product in nano technology, biomedicine, biotechnology, 3D printing, and nano fabrication and semiconductors. He has been a co-founder for multiple startups, including Azul, Nanosphere, and much more. He's traveling all the way from Chicago to share his experience. Furthermore, we would like to extend our sincere appreciation to Beta Lab for sponsoring this event and for King Saud University for hosting us. This collaboration and motivation within the ecosystem are truly inspiring. Ladies and gentlemen, we promise not to keep you longer. We are eager to hear from D.Shad, his insights, and the wonderful experience that has been through it. D.Shad, the mic is yours. Welcome, D.Shad, please.
Okay, great. Thank you for the nice introduction. It's really a pleasure to be here. I want to thank also His Excellency Aswaha and the RDA Authority for making this all possible. I came here, I don't know, 7 months ago or so, and for the King Faisal Prize, which was just an incredible event, and started a relationship that continues today and continues to grow at a really rapid pace. As she said, I'm involved in a lot of things at the university. One is the development of the field of nanotechnology, but the other is the use of many of the discoveries and inventions that we make there that can be translated into commercial products and the basis for a whole wide variety of startup companies. And what I thought I'd do today was tell you a little bit about how we do that at Northwestern. And then keep it somewhat informal. You guys can interrupt me if something's not clear, and I'll try to clarify, but then open it up for questions and exploration after that. And so I started what was called the International Institute for Nanotechnology about 25 years ago. And this was a big deal back then because I was just starting out as a young professor at Northwestern. And the modern age of nanotechnology was just getting going. And kind of like what we're doing here in terms of some of the companies that we're bringing to Saudi Arabia, I saw an opportunity. And the opportunity was to, one, define a career in nanotechnology, and the other was to define Northwestern in the area of nanotechnology. So my view was that this is a new field, and we were starting at ground zero, and the entire world was starting at ground zero. So we had an opportunity to get up to the top fast and to stay at the top. And that's unusual, right? Because oftentimes you're defined by your chronology, when you start doing things and who's ahead of you. In the US, oftentimes the East Coast is ahead of you because they started a long time ago, and West Coast has played catch up to some extent. And the middle is there kind of in between. And so I convinced the president at the time that this was the right thing to do, and that he should invest, and we shouldn't tiptoe in, we should go in aggressively and use this to define Northwestern University. And today, I like to say they call Northwestern NU not because of Northwestern, because it's Nanotech U. Because this had a huge impact. Nanotech is a field that impacts all disciplines, all of science, all of engineering, and much of medicine. And as a result, it across the stage can have an impact in all of those disciplines and create opportunities for everybody that's involved. And so we started out small, and today, if you look at what we have multiple operations. We have an Evanston campus, which is in the north, right on Lake Michigan. That's where the headquarters are. But we also have a very strong presence in the medical school. We built the world's largest, or the U.S.'s largest, biomedical engineering operation. Lots of ties into medicine, as you're going to see. And then at Argonne National Laboratories, which is kind of our CAXT, we built a Center for Nanoscale Materials that was all very tightly connected. And then we began to build out from there and built what I call or refer to as the IIN. And so you can see kind of the history here, and I'll point to this. It was zero back when we started. It's raised over $1.2 billion in terms of infrastructure geared towards state-of-the-art education, research, and commercialization. And as I said, it's impacted almost every group of folks that have been affiliated with it since the beginning of the Institute. And it's become an international hub of excellence. So a lot of the interactions we've established are not just local, I mean within the Midwest, not just national, but international. And it became a big draw. And that was kind of my thinking in doing this. We want to build something that attracts talent. Because I don't care what you're building, whether an academic enterprise or a commercial enterprise, if you've got access to talent, you can do things that others that don't have access to talent can't. And so today, it has become a hub and a magnet for both talent and also commercialization. Because many of the things that were developed were not just scientific discoveries, new ways of thinking about doing things, but new capabilities came out of those new ways of thinking about doing things, new technologies that impacted a lot of different disciplines and opportunities. And so if you kind of look at how we separate these, you can see across the board, part of it is about developing tools that allow you to do nanotechnology. There's a business in that. I'll talk a little bit about that. Commercialization capabilities and startup companies that have come out of that. How do you make things small? How do you manipulate matter on the nanometer length scale? How do you do that routinely? That's the kind of the heart, the bread and butter of anybody doing this type of research. Environmental solutions. I always like to say the fastest way to new materials, and materials drive everything that we do, is through miniaturization. And the reason I say that is that if you take any material and shrink it down to the nanometer length scale, it will have new properties. You take the gold and the necklaces you wear or the rings that you wear, you shrink that down to the nanoscale, it's no longer gold in color. It can be red, blue, green, any color of the spectrum you want. Exact same material, but different structure on that scale, different properties. Different properties create new technological capabilities. Food and water safety, energy solutions, and of course medicine, which is one of the things that we're really heavily involved with, because I think that much of what we're doing has a chance to redefine how medicines are made, how we actually make really potent capabilities in terms of treating different types of disease, how we track disease in unique ways. And so many of those types of tools are going to transform the way medicine's ultimately practiced. So you have all these people that are in the Institute, and we now have literally hundreds and come from all parts of the world. I say we don't discriminate with respect to talent. We'll say if you've got talents, bring them to Evanston. That's kind of the mantra. That's also led to an enormous number of inventions, over 2,000 inventions to date and growing. And many of those inventions, as I said, become the basis for commercial products. I always thought from a building the center standpoint or the Institute standpoint, that's kind of our intel inside. If we have technologies that are used all over the world and they trace their roots back to Northwestern, that's a big win for the university. That was also one of the ideas behind all of this. Enormous amount of venture capital invested in now over, I think, 32 different companies. And some of the more recent ones are shown here, and I'm going to give you a few case studies in terms of how we made discoveries that led to technologies that could become the basis for different types of startup companies. Okay, so these come from, what I talked about, come from hundreds of researchers. What I thought I'd do for today is just pick a few examples from my lab that have led to the basis of new scientific discoveries and technologies that could become the basis for different types of startup companies, some of which we are commercializing and developing with the kingdom. Abdul Rahman's up here. He's been a huge proponent of what we're trying to do from Beta Labs. and ultimately built the capabilities, or helping us build the capabilities for Flashpoint Therapeutics, and also another company called Azul 3D, which is a high throughput 3D printing company. We have other companies in the clean energy space, including Matique, and a fourth company I'll talk about. Matique is Mat IQ, where matter meets intelligence. I'll talk a little bit about that. It's a big AI and clean energy materials discovery play. But then TerraPrint, which is a tool-based company that enables much of the technology I'll talk about as well. So four kind of different companies, four different types of strategies in terms of development. One common theme, which is they all get their birth in the Institute, and we use this ecosystem to ultimately develop the capabilities that I'm going to share with you today. So the two discoveries that are important in my lab, one is what we like to refer to as spherical nucleic acids. So DNA is probably the most important molecule ever discovered by scientists, ever synthesized by chemists. It comes in a form that probably everybody in this room knows a little bit about. It's a linear duplex type of architecture. We know the A's, the T's, the G's, and C's that define the genetic code. That's how it occurs in nature. In nanotechnology, we can make it into a new form, a globular, spherical form, by taking tiny little particles and short snippets of DNA and modifying those particles so that it's presented on the outside. That spherical nucleic acid is only made through chemistry, only made through nanotechnology, but it actually completely changes the way the most important molecule ever discovered by scientists interacts with life. And those new ways lead to new ways of tracking and ultimately treating disease. And so that can become the basis for commercial types of products. It can become the basis for startup companies. So that's going to be number one that I'm going to share with you. The second one is what I like to refer to as the world's smallest printer. We developed a technique called dip pendant lithography that allows you to use something called an atomic force microscope to literally draw out molecules onto a surface. Just an academic curiosity 25 years ago. Today, we now have built commercial capabilities that use literally 11 million of these tiny little tips, like little fingers, to control where chemistry occurs on surfaces. And it completely changes the way you think about a lot of issues in printing, a lot of issues in 3D printing, as you're going to hear about, and a lot of issues in terms of materials discovery, because each of those little tips can be viewed as a kind of set of hands chemically synthesizing materials in parallel. So you now can make materials faster than have ever been contemplated before, and that opens up great opportunities in terms of materials discovery. And so here are a whole set of technologies that have come out of this, ranging from new medical diagnostic tools to new therapeutics. I mentioned desktop fabs, so imagine instead of having to go to a fab to make a microelectronic device, you can make it at the point of use. Instead of having a clean room, you can do it out in the open. These types of tools enable that. The world's most parallel synthesis tools, and then the world's highest throughput 3D printing capabilities. I'm going to show you a tool where I could print you in a couple of hours, which is just warp speed for 3D printing. So any sort of configuration you'd like, complexity and scale that's never been seen before. Okay, so the first example, spherical nucleic acids. The way this worked is we took tiny little particles, first made of gold, now made of liposomes, little fat globules, could modify them with DNA or RNA, and you could turn them into these types of structures. Why are these so interesting? This is kind of the aha experiment. Turns out they have all sorts of different properties. They're more stable than normal DNA and RNA, so if you're developing a therapeutic, that means they're going to have a longer therapeutic lifetime. They'll actually engage and bind with complementary targets more effectively, higher affinity than normal DNA. That means if you're trying to fish out genetic signatures of the disease, you can do so at lower concentrations. So all of these properties, fundamental properties, lead to new commercial potential or technology capabilities. But this is the one that's just staggering. So normal DNA and RNA will not enter cells, your cells. And there's a reason for that. Your cells don't want a lot of foreign DNA freely moving in and out of them, because that can throw in a wrench that messes up the machine. Unless you're trying to measure things in cells or manipulate what goes on in cells to develop new therapeutics. And so we discovered, it took us about 10 years to run this experiment, because you're taught these guys will not go into cells. You can see if I put a little red dye on here, there's no uptake of the DNA. I take those same DNA strands and arrange them in spherical form. Now they not only go in, they go in better than anything known to man. They're actively recognized by the cells and internalized. And so the reason I said it's an aha moment, you say, oh, now I can just through structure get large amounts of genetic payloads into cells, make measurements inside cells that couldn't be made before, and also manipulate what goes on in cells and develop a class of medicines based upon that capability. And that became the basis for a wide variety of different types of technologies. You can kind of see, this is the odyssey. We go back to 1998, first time we make, well, actually it's 1996, spherical nucleic acids. And along the way, we begin to think about how we can first use them for diagnostic tools. That led to a company called Nanosphere, which was a medical diagnostic company we built up, took public in about 7 years. It was bought by Luminex, which was bought by Soren. That technology now is the basis for a lot of the genetic testing that goes on around the world. That medical diagnostic system I want to show you is now part of over half the world's top hospitals. We then began to ask, well, how can we then use this to make measurements inside cells? And we invented a technology called nanoflare technology that allowed us to begin to make measurements inside living cells so that I could take a sea of cells and feed them these types of probes, and only if a particular genetic signature was present would they light up. Why would I like that? Well, let's say I wanted to look for circulating tumor cells. I wanted to pick the needles out of the haystack to find the cells that had aberrant DNA that I could then study and then ultimately use to understand that disease or develop new types of therapeutics. That led to a company called Arasense, which commercialized those technologies with Millipore and now are used all over the world. And then all the way to a new company called Flashpoint Therapeutics. So we just started here in part with investment from Beta Labs and a wide variety of investors around the world. And this is a company that's poised to completely transform how we develop immunotherapies, vaccines for cancer, for example, how we create really potent therapies that can ultimately treat disease in unique ways. And I want to show you a couple of examples of that as well, because I think it's really exciting and has, as I said, a chance to really redefine how we think about developing medicines. Okay, so this is the first system, the Verigene system, as I said, used in over half the world's top hospitals. We use those types of particle probes effectively to fish out targets of disease. Everything living has a genetic signature. So every viral disease, every bacterial disease, every one of you. If I know what those signatures are, I can develop probes for them. And if I have a sample, blood, saliva, or urine, I can treat that sample, release those DNA signatures, latch on with particle probes, and then I can use a chip that has different spots of DNA to sort and pick out the particular signatures that are present and bind them to a chip. These particles, this is one of the few examples where we used gold particles as the core of a spherical nucleic acid. Remember I said if you shrink gold down, it's no longer gold in color, it's red in color. It's bright red, it's like blood red in color. It's remarkable, very good dye. That gives you then a strong signal in that particular quadrant, but that's not strong enough. Turns out you need to be able to fish out really, really low concentrations. The other thing that's important is that gold is catalytic. So I can develop these chips with photographic developing solution, just flow ordinary photographic developing solution, the old days when we used photographic developing solution. And anywhere where the gold particles are, you plate out silver. And now I can use that as a waveguide, and when I bring light through here, anywhere where I have these silver gold blobs, light scatters off, and this is the type of signal that I see. So here's a panel assay for HIV, Ebola virus, smallpox, and hepatitis B. And you can see in the panel assay, we've got the test run 6 times over. So all positives here. So the signatures were there. And here you can read this chip very easily, right? Hep B here, smallpox, Ebola virus, HIV here, and then every combination thereafter. So this was a really simple and fast way to ultimately track disease in a unique way. And so that became the basis for this company called Nanosphere. We got the idea, raised venture capital to begin to go after that, began to develop the first commercial tools, and then ultimately built that capability out over the course of about 7 years. We took the company public, ultimately built it out, was, as I said, acquired by Luminex, and now is used over and over and over again all over the place, including, for example, for tracking diseases like COVID. So dramatically changed the way disease is detected at the point of use. So the great thing about this system, you don't need a remote capability. You can basically do it at the point of use in hospitals, even in the doctor's office was, you know, the early case we made. It's primarily used though in hospitals. Okay, what else can you do? Well, these signatures are amazing. You can begin to build particles that can go into cells, and we can add little things called flares that where the particles are designed to bind to a specific type of target mRNA that's present. Fancy way of saying we can track cells based upon genetic signatures. So let's say we wanted to look for circulating tumor cells. We could go after an mRNA that's overexpressed in cells that are associated with cancer cells. And if it's present, when that happens, they bind and liberates these fluorophores, and the cells light up. Okay, and so if I take a group of cells and I feed them these types of particle probes, they'll only light up the cells that overexpress this mRNA. And now I can fire them through something called a flow cytometer and literally pick out the needle in the haystack. And I not only can pick it out, I can pull it out and keep it in live form. And that was the only way to do this. There was no other way to do this. So that allows me to not only diagnose, let's say for breast cancer, which we developed a type of test for, but I can collect those cells and culture them and then begin to look at how they respond to specific experimental therapies. So this is not only a powerful way of studying disease and tracking disease, but ultimately treating disease. I always say the first step to a good therapeutic is a good diagnostic. And so we're kind of developing all of these suites. This became the basis for Aurasense, which commercialized these, made over 1,600 different versions of these, and sold them and used them all over the world. And you can see what they're used for: circulating tumor cells, high throughput drug screening, even in animals. You can look at, for example, in the skin. This was in rabbits. We were working with a group from Singapore, asking the question, could we begin to differentiate cells that were susceptible to keloid formation? They overexpress a certain type of RNA, and we could begin to track who had those types of cells and who didn't, and then, of course, think about how we could use that to intervene with different types of therapies. Let me skip this for a second. Let me go to the third example here. Nanotechnology is revolutionizing immunotherapy. And with immunotherapy, I include all vaccines, right? We all know we've just gone through COVID and the importance of mRNA medicines. It's kind of interesting if you think about the way vaccines have been developed to date before we had the mRNA vaccines. This is going to be viewed as the Middle Ages, 100 years from now. Kind of crazy when you think about it. When we develop a vaccine, we effectively look for two components: something to stimulate your immune system called an adjuvant, and something to train your immune system to selectively eradicate foreign invaders or cancer cells if you're developing a cancer vaccine. And that's an antigen, so oftentimes a peptide type of signature. And the old way of doing this is what I call the blender approach, is grabbing those components, mixing them, injecting them, and hoping that your immune system does the work that you're on. And remarkably, that actually works for a lot of applications. But as a chemist, at least, and a nanotechnologist, I thought there has to be a better way. Well, we know there is at least another way, and certainly a better way in treating certain types of diseases, like, for example, COVID, because I can use nanotechnology to begin to compartmentalize the components that matter, or to deliver signatures like mRNA that make you generate the signatures that matter, a peptide unique to COVID, for example. And the beauty of that is I'm beginning to use structure to put together these components in a way that increases their efficacy. And so at the very least, when I put these guys together, I can deliver mRNA, and I can use that to get your cells to be stimulated, and I can also get your cells to manufacture effectively a specific protein of interest associated with COVID, for example, to create a response. And so this is kind of the first step towards showing that these types of structured medicines are going to play a big role, and nanotechnology is going to play a big role in terms of making them more effective. Now, crash course in terms of immunotherapy or vaccine development, right? So don't get caught up in all the details here if you're not an aficionado. but you can impress people at the next party you go to, telling them I know the basics of how vaccines work. As I said, I have two components, an adjuvant and an antigen. Typically mix them, and what happens is you're trying to get these cells called naive dendritic cells in the case of a cancer vaccine to pick them up, and those cells are going to process these signatures. It's going to activate the dendritic cells and create two signatures here on the surface. One that begins to allow these types of cells to train killer T cells that dock onto them, to go out and selectively find certain cells. So you don't want your immune system to kill everything. You'd like it to only kill specific cancer cells. So you pick an antigen that is specific to the cancer cells that you'd like to eradicate. The way these get processed has to depend upon structure, right? So there's a very simple hypothesis that if I present them in different ways, I should be able to get different types of outputs and different efficacies. That's kind of a premise that's really important for Flashpoint and what I'm going to tell you about with respect to structural immunotherapy. Once these signals are generated, T cells dock, they mature, and then ultimately go out and find only the cancer cells that have that specific signature and then lyse them. That's the basic premise behind creating a cancer vaccine and training your immune system to eradicate cancer, as opposed to using more conventional forms, for example, of chemotherapy. Well, how do spherical nucleic acids come into play? Turns out you can make spherical nucleic acids out of DNA that's known to stimulate your immune system. There are specific sequences that are known through the study of bacterial and viral infections that you can incorporate into a spherical nucleic acid, and now you have an adjuvant. Well, we then took that concept and began to form a company called Execure and take that concept to the clinic. Raised a tremendous amount of money to set the stage for that. And I'm going to show you a trial that I think is really quite exciting. This is where we were using a drug we call Cavro. It's what's called, for the aficionados, a TLR9 agonist. Stimulates your immune system. And in this particular case, the idea was not to create a vaccine, but just to create an adjuvant, something that would boost your immune system. And the hope was that it would work with things called checkpoint inhibitors that are miracle drugs for a small percentage of the population, but a large percentage doesn't respond well to them, to effectively treat two very difficult forms of cancer. One is Merkel cell carcinoma, a deadly form of skin cancer, and the other is squamous cell carcinoma. And we took this into the clinic all the way to phase 1b/2 trials. And showed some pretty remarkable results. That we saw a target tumor shrinkage in 37% of the patients, and in certain cases we had complete responders. And so here's actually a patient. This is a 92-year-old male. They've, by the way, failed every form of therapy, including immunotherapies that are out there. This person, of course, signs up for this trial, takes our drug along with Pembro. And this is what it looked like with the treatment of just Pembro without our drug, or no treatment whatsoever. This is what it looks like after treatment. Really remarkable. I mean, the patients are riddled with tumors. They're completely gone. They melted away after being treated in this manner. And so this is, to me, the first sign that this idea of structural immunotherapy is actually going to work. But we don't want to stop there. We want to begin to develop a whole series of vaccines by basically asking the question, how can we take those components that define what I referred to as the blender approach, put them together in the spherical nucleic acid form to create really potent medicines? And that's the basis behind this new company called Flashpoint Therapeutics. We just raised a seed round of $10 million and are off to the races. We're going to bring a lot of these medicines to the kingdom and begin to develop the company and also the technology around the world, but also heavily through the collaborations that we've been establishing within the kingdom. And the basic premise of structural immunotherapy, or what I like to refer to as rational vaccinology, is that structure matters. That if I look at the components alone versus in a particular format, architectural format, there must be formats that are either more or less efficacious. And if we can figure out what those ones are that are efficacious, we can begin to really raise the bar and capabilities with respect to immunotherapies in general. And so to give you a feel for how powerful this is, here's an example of a structural immunotherapy in play. In this case, we're looking at HPV cancer model, human papillomavirus. And we have our peptide here and our spherical nucleic acid or linear nucleic acid adjuvant. This is the blender approach. You can see in blue here, these are animal studies where we've intentionally given this cancer, and then we treat with these particular components. And you can see with the blender approach, there's almost no effect at all. Tumors grow out of control. If you follow the blue here, animals die very rapidly. I look at 3 different forms. The spherical nucleic acid platform allows me to adjust the different ways that I can present these components, right? We have to figure out what the right presentation is. And you can see here we have an encapsulated form where we put the antigen in the core. We have one where we put it between the adjuvants, and we have one where we put them on the adjuvants. Look at the data here. So this has almost no effect. It's very tiny, maybe a little bit increase in terms of survival. This one looks pretty good. This is the purple, both in terms of retarding tumor growth, but also animal survival. But this one's amazing. So it's almost completely curative in a good fraction of the animals studied. Okay, so that data set alone tells you that structure matters. That I can use nanotechnology to restructure immunotherapies to create really powerful forms. And it really is kind of a charge to the whole field to reconsider how you think about making vaccines. And that's what we're bringing through Flashpoint and all the things that we're doing with Beta Labs and the different hospitals within the country. This is the reason why. That structure is a privileged architecture. It has unbelievable benefits in terms of how we get the components into cells, how we increase the activation of the cells, how we control antigen presentation. That's the important part about training the T cells. And then this is the really important one, 6.5 times increase in tumor cell killing by T cells. Okay, so we think this is a really powerful way of thinking about developing medicines going forward. And I don't say that lightly. That's not just a one and done type of thing. 9 different cancer models. There's not a single exception where if I compare this to the structured form of the immunotherapy, I move from the down the ineffective to curative axis. I will not always get to curative, but I will always get to something that's better than what I had, for example, with the components alone. And so we think this is about redefining how you think about developing vaccines in general, but also immunotherapies in general. Okay, let me show you the last examples with the printing capabilities, because I think they're kind of interesting too. Different types of sectors impacted, going beyond medicine, but medicine's also impacted as well. So remember I referred to dip-pen lithography back in the late 90s. That was a big deal. People referred to it as the world's smallest pen, basically using these tips from an AFM, which was used initially to get topological information about a surface. That's what they were invented for. We transformed them into writers, where we could put chemicals on the surface of the tip and locally control chemistry that occurs underneath. We initially thought this is going to be a way to draw out electronic circuits and effectively to develop a type of desktop fab. And in fact, tools were developed that allow you to do that and are used all over the world, including in Saudi Arabia, to do that as well. Primarily research tools, but really powerful tools that allow you to miniaturize electronics very rapidly. But it turned out that this laid the basis for completely transforming how we think about doing chemical synthesis and maybe redefine the entire chemical industry, as you'll see in a little bit, and certainly the clean energy industry. But we were thinking really, really small. But over the course of 20 years, 25 years, we found out how to actually make the world's largest 3D printing capabilities using concepts related to this. And so that's the last little story that I want to share with you as well. And that became the basis for a company called Azul 3D, which Beta Labs has also invested in along with other investors around the world. Okay, so again, with all my kind of pursuits, there's a timeline. I guess it's a sign of my age. You know, we are developing at the time this idea of miniaturization, primarily for electronics. There's another guy up at Harvard, a guy named George Whitesides. I think he might have also won the King Faisal Prize too. He was developing parallel printing tools, micro-contact printing tools. The basic idea was that you could make an elastomeric stamp with little micro features on it, and you could use that to transfer chemicals to a surface, transfer effectively a pattern in one shot to a surface. A lot of people thought these were competitive. They're not competitive. They're just highly complementary. We're high resolution, small area. He's low resolution, large area. We began to think about, are there ways to take and merge the two and capitalize on the strengths of both of these and eliminate some of the weaknesses? And we came up with something called polymer pen lithography, which is really quite interesting. The basic idea is with an AFM, tip is used kind of like a spring to measure, as I said, topological features on a surface. And it has something called a cantilever that's used to do that. We thought we could take that spring and put it in the tip if we made it out of an elastomeric material, rubbery material, something like PDMS. And then what we could do is build a mold where we could make a lot of wells that were pyramidal in shape and use that to build polymer tips that could be almost unlimited in terms of number and area. And we then could insert that into the same instrument that was used for doing atomic force microscopy to locally control chemistry underneath, but now in parallel. So instead of having one tip doing chemistry on a surface, we can have thousands, hundreds of thousands, even millions of tips. So it's kind of, think of having, you know, 11 million people now locally doing chemistry on a surface over large areas. That's important. These ideas then became the basis for commercial tools, things we call the TerraPrint series. A company called TerraPrint was started, actually by myself and my son and one of the guys who invented this technology, a Russian named Andrei Ivankin, who actually was here for the King Faisal Prize, and quickly laid the groundwork for thinking about doing this in a variety of ways. One is where we could locally control chemistry by depositing molecules, or where we could use the pyramids as light pipes. And we could take the same type of device that's in a projection TV and take one beam of light and split it into 22,000 beams of light. Now independently dance the light off the back of these pyramids and control the chemistry with light on the other side. And so that makes every one of these tips independently addressable. That now is the basis for commercial tools that I'll share with you. But we then quickly began to think about this not as a fabrication tool, but as a synthesis tool. And that enabled this concept of mega libraries, where I can take a chip, much like a gene chip, but instead of making spots of DNA or proteins, I can take a 2 by 2 centimeter chip and make 56 million distinct materials, different combinations of elements in the periodic table, all positionally encoded. And now I can begin to look at them in mass to find the materials that matter for a given application. I'll show you how we're using that to find new catalysts that can take feedstocks like CO2 and convert them into high-value products. Take water and split it and generate hydrogen uniquely. And in fact, electrify almost anything using this as a way to find the materials that allow that process to occur under acceptable conditions. Okay, so these are the tools, just to give you a feel. These are sold all over the world. We started a company called TerraPrint. Many ways to do it. Many times we go out and raise venture capital. In this case, we raised zero venture capital. I actually funded it and with a loan. And within the first year, they sold enough of these that they paid everything back and they're off the races and now beginning to populate really many of the research labs around the world with these types of capabilities. These tools allow you to do what you would normally do at a fab at the point of use, whether you're thinking about prototyping for electronics and microfluidics, biopatterning, polymer nanoprinting, or as you're going to see, parallel synthesis in really fantastic ways. You can see some of the outputs shown here. Anything I can digitize, I can then transform into a pattern using the capabilities that I was referring to. And so here you can see electronics. I can prototype electronic circuits. These are patterns of proteins that allow me to control how cells are positioned on surfaces, the different types of morphologies. I can use that to study how cells function. I can begin to develop starting points for developing cellular-based diagnostic tools and lots of other capabilities. And here's a mega library where I have a few, 2 minutes. I have a chip that has 56 million unique materials on it. So this takes you to the next company, right? Matique, MatIQ. This is a way to completely accelerate how we find materials that matter for many different types of areas. In this case, the clean energy space. But what I want to point out is that this is not only a tool that allows you to make materials that might be really important for whatever it is that you ultimately want to use a material for, but these represent the largest source of really high quality big data that can be used to empower machine learning algorithms. And so we're using these types of capabilities to find catalysts that allow us to think about tackling the CO2 to high value type of product conversion, allow us to take and replace, for example, iridium in the case of the OER and water splitting types of electrification systems. Let me skip this through here. Okay, last thing I'm going to show you is the 3D printing capability. So everything's been about small. This is about large. So one of the things that we were looking at was when we were making those tips and trying to use them as light pipes to control chemistry, we said, hey, we could affect polymerization. And that's used in 3D printing, so we could make the world's smallest 3D printing capabilities. The problem was that when we did that and we tried to take a vat of polymer and polymerize underneath the tips, we could polymerize, but the polymer would stick to the tips. And so what we thought about was, how could we stop that from occurring? So we took a fluorocarbon oil and put it on the tips, and that fluorocarbon would keep the polymer, when it forms, from sticking to the tips. And then we could make higher quality types of structures and begin to print continuously. Then we said, wait a second, we don't need to do this just on the nanoscale. We could actually do this on the macro scale. We could put that fluorocarbon oil and just use a projection type of TV, a digital micromirror device, to project an image. And as we move a plate up, we could print first the generation, the first layer of an object, and then continue to print the object as we go from bottom to top. And the fluorocarbon would keep the part from sticking, and therefore we could have continuous printing and we could be fast. The problem with that was that when you go big and fast in 3D printing, you generate a lot of heat, and the parts start flaming. They start literally catching on fire. So then we thought about, well, we could take that oil and we could circulate it through, and we could use it as a non-stick layer, but also as a way of pulling the heat out and moving it through a chiller, wrapping it around continuously so that we could solve one of the biggest problems in 3D printing. And now we'd be unlimited in terms of size with respect to printing. And so here you can see these are propeller parts. In this case, how can I? Did it go? There it goes. There you can see it printing right here. These are, almost unlimited in terms of capability. We're now building 3D printing capabilities where we have 64 projection types of systems, DMD devices, all printing in parallel over very, very large cross-sectional areas. And so this is really exciting because it completely transforms how you think about what you can make with 3D printing and what you can ultimately manufacture. And so whether we're talking about medical, automotive, industrial, sports equipment, even prescription lenses can be made using this, because the continuous nature of the printing capability allows us to print a really high quality product as well. And so that's kind of the basis for this company called Azul 3D. And these are the types of printers that we ultimately have commercialized, are now going all over the world as well. But this is really just the start. This type of capability will expand dramatically as we move forward. I think I'll stop there because we're running out of time, and I'd be happy to answer any questions that there might be from the audience. Okay, so he's asking what makes the spherical nucleic acid so special? cells. In terms of the clustering of DNA, it's not so much that it's spherical, it's that we've come up with conditions to cluster the DNA and RNA on the surface of the particle. And there are things called scavenger receptors that recognize clusters of nucleic acids. And we don't recognize linear nucleic acids very well, but they do recognize things like G quadruplexes and spherical nucleic acids that facilitate the movement of these types of structures into cells via a process called cationic cytosis. And so it's really the clustering, not so that the spherical is more descriptive. It's the high density. In fact, if you lower the density of DNA on the surface of the particle, you'll see the DNA go down. That's all been studied in great detail.
Well, Chad, thank you for giving us an aha lecture. For many reasons. Well, I'm easy leadership and the vice president. Ironically, we've been looking at 4 universities in the last period for our transformation. So we started alone, and now we're a independent non-profit. We've been looking at Toronto with a volume of students. Evolution in the last couple of years in terms of the high-tech powerhouse, and expenditure of capital. You know that we have one of the healthiest research budgets anywhere in the world now. On the AI to give us more money. But one of the most challenging things in the creative process is how to spend money. Only how to get it. So, how do you see, and what's your, like, in a nutshell, advice for us at KSU?
Great question, fantastic question. So first of all, I will mention, I'm happy you said that, because Michael Pretzel is one of the most accomplished people in the energy space, working at the Welsh Conference. Or Europe, watch what you do. That's the rise of Northwestern is like no other university, certainly in the US. So I would like to say that I feel like we've done things the right way. The question I think is really important because my experience has been in the academic sphere is a lot of people like to collect money, sit on money. So I don't think that makes sense. I'm not a believer. I'm a believer in investing that money and deploying it properly. I've always told all my bosses, the president, and everybody else that when I ask you for resources, when I go spend them, I'm going to do great things with them, then I'll come back and ask you for resources. And the key is to not have a lag time. So to your point, I also follow some of those universities. I lived in Toronto, and I moved Ted Sargent and Shana Kelly, the biggest clean energy person, one of the biggest biotech people out of Toronto, to Northwestern 2.5 years ago for that very reason. He was go after best, move them, give them great opportunities, give them resources, get out of the way, and tell them if you spend this properly. It means venture capital. Learn a lot from venture capitalists. Don't keep throwing money at credit venture capitalists. They keep making bad investments and take your money, put it elsewhere. Keep making good investments. Give more money. Opportunity. So I think the key is to get people out of the money hoarding mentality, to hold people accountable. Don't just tell me how good you are. Tell me how you're going to get better. How are you preparing yourself? What are your strengths, but also what are your weaknesses? You can't solve a problem if you don't recognize a problem. Big believer in that. So I know how to chess beat like the best of them out there, but I also like to focus at home on where are our deficiencies and what do we have to fill to get better. You never rest in your laurels. It's such a competitive business that I don't care how good you are. I came out of MIT. You can squander reputation, you can squander opportunity in a really short period. It can take you decades to build it up, and it can take you years, a few years, to tear it down.
Policy change.
are they a high enough quality? Of course they were. If you view this as a net benefit, let's go forward. That turns out to not be a simple thing to do. Because traditionally, they've always hired at the department level. Everybody gets really territorial. If I give up to this to that institute, then I'm never going to get this back. Whereas I say it's the opposite. If we get something really good that impacts our institute, it's going to impact your department. And we'll make a case for going and asking for more.
Chad, I'd rather not ask you any more questions because I would go on and on. I have one more technical question. These spheres are not cancer genetics. Do you see attaching higher complex or more complex structures to it, for example?
We have CRISPR-Cas9 published on this. So Cas9 loaded up with a guide RNA, target RNA, transformed it into spherical nucleic acid. You can even express it in a way that you've got endosomal release, a type of protein, nuclear localization signals. And so now you have one construct that is all put together, the whole machinery is put together, and will go into almost any cell type. So you overcome one of the biggest limitations in CRISPR. Chemically attached to no spherical. Yeah, we have. You don't get cellular entry. So the cellular entry is key. So he's asking a really good question, a very important control type of experiment. If you just attach the two, that would not be the molecular approach that you did connect it together. And that might provide some advantage. Actually, we can go a little bit of this. But the reason these work so well is the spherical architecture, the high density nucleic acid architecture gets you into cells. That's important. And you set up the ratio of adjuvant to antigen the way you like ahead of time. Control slope chemistry. They all go to the same cell type, which is important. So that's controlled in the experiment. And then the signal, the processing, remember the signals that are generated in the case of the vaccine, the timing is markedly influenced by the structure that you use. And so what I often say in a longer talk or one nuts and bolts of this is that structure is really important. The timing may be everything. The structure is the root to controlling the signaling kinetics of vaccines. So answer your question, you get a little bit of the way there, but you don't get all of it. And a big part of it is actually getting the cellular data for a potent response. So he's asking, so when we make, let's say, a library of 56 million new materials, we will take spray guns with the precursors, 4 different elements, for example, salts, if you're going to take metal salts. We will spray them across the array of tints. So pure 1 here, pure 2 here, pure 3, pure 4 here, and the gradient of everything in between. Now you put that in the instrument, you come down, deposit a small feature, and that feature's composition reflects the spray gradient that you started with. And so there's something they call the discovery factor that allows you to rapidly do this and build these massive libraries. But think about it. I just want you guys to think about how important that is. So 56 million materials in a 2 by 2 centimeter ship, internet materials. The world to date, beginning time, has made and characterized less than 1 million. Types of tools are going to have to create an inflection how fast what types of materials can be discovered. I cut it out, and we've already found 5 world records. The world record for fluid mission for halide perovskites. We found a replacement for iridium in terms of water splitting, the OER reaction. All this electrolyzer type business is based upon the use of iridium. A lot of people don't want to use iridium because it's localized in one part of the world, and there's just not enough iridium on the planet to supply the projected world's needs. We found a 4-element replacement. We just submitted that to Science Magazine. By doing this massive 2.25 million material suite and looking at all of them in terms of their electrocatalytic capabilities and mass, and again, a structure that nobody could have predicted, you can find very rapidly these techniques. And that's not even using AI. So they're then taking local space and ask, okay, what, how good was the machine, and where among all of these kind of general possibilities you're going to see a feedback back and forth for quite some time. I think about 5 to 7 years from now, you're going to see predominantly AI as the value proposition here, and very tool percentages of affirmation. Here, actually, I'll answer part of the answer your question too. That's kind of a policy issue. So I would like to say, when I first started, if you said you were going to develop a company and raise money and go out and build a startup, you were dancing with the devil. Today, they give you medals for it. Right? So the change in view has been extraordinary. Now, I think you'd be careful to have a healthy emphasis. At the university, first and foremost, it should be about scientific discovery. Look at every one of the inventions we have here. It's really geared towards how do we make things in a new way? How do we, what's the difference between linear and spherical in terms of behavior? A lot of fundamental exploration that led to discovery and invention. But then you need to train people to have a mindset to look and say, hey, here, that's something I learned there. This brings a new technological capability. That actually have value. And to encourage them. But I don't think you want to turn everybody at the university into a business person. I think you want to interface, you want an ecosystem of people that are really far on this side of things that really like the translation. People over here that are like the tinkers, and have them work together and kind of create alliances that keep their eyes open and look for those opportunities. And you've got to mesh with the government, the capital community, and create an ecosystem to create lots of shots on goal. So there'll be a lot of failures. We can reduce the amount of failure. We can not eliminate all the failure by doing this exercise.
Thank you. Appreciate just to follow up with this question. At what TRL level should research be executed for the team? At what TRL level?
I don't know the lingo in terms of TR level, but I can tell you readiness. I guess, but again, when we hire somebody at Northwestern, we don't care about whether they're going to commercialize things. We just want the smartest people in their particular area. Have them trained and develop their science. And then we have this ecosystem where many people have done startup companies and know how to go. So they know that exists. And as they get more interest in that area, they'll either be careful going out and transfer it over to them and say, hey, you run with it, or they'll work with them to do it themselves and they'll learn. But I like to think that at the university level, you want to start first and foremost with the best scientists and the people. And that's the foundation. Basic science is ultimately the foundation for everything technological. But build the whole ecosystem so there's a range of people and experiences where people that are just starting can begin to interface with them and find the path. In my case, I started at Northwestern. I did not do it. I started on over at business school. Kellogg just started knocking on doors and saying, look, I think I have this idea for medical diagnostics. Anybody can help me here. They have people that did hypothetical business plan and began to kind of develop that and put it into motion. I think we have to go, right? But anyway, you guys can reach me by email. I'll try to respond as quickly as possible. Thanks so much for having me.